Let's briefly talk about Earth systems and proxy data. So, Earth systems are those big, big spheres where we talk about processes happening. And the first is the geosphere. So, it's the part of the Earth that includes all the rocks, the minerals, the land, everything that's beneath the ground and our feet. So, mountains and valleys and soil. It's all of the geology that we study, the geosphere. The hydrosphere includes all of the water on Earth. So that's fresh water and salt water, rivers, streams, glaciers, and even groundwater. So it's all of the water systems. And we think of the hydrosphere and the water as being fairly reused within our Earth system, within our atmosphere. The atmosphere is the layer of gases that surround the Earth, and it comes all the way down to the surface of the Earth. So it's what we breathe. The atmosphere protects us from sun's harmful rays, and it contains numerous different gases, oxygen, nitrogen, and water vapor. The biosphere is all living things, plants, animals, even tiny microorganisms. So it's really where it's the area of life where all living creatures are. So that would be the biosphere. And then there are sub areas, like the cryosphere refers to Earth's ice in all of its forms. It's from the Greek word cryos, which is icy or cold. So it can be the snow on the ground, snow on lakes and rivers, frozen ground, permafrost, ice shelves, icebergs. So it is sometimes thought of as part of the hydrosphere. But there are people who study the cycling of the cryosphere as well. So a proxy record is their data sets of preserved physical characteristics of the environment that stand in for direct measurements. So a proxy record tells us things about our environment and our Earth in the past. So it can approximate something else. So it's most commonly, we refer to proxy records as natural recorders of the past climate. Examples of that are tree rings and ice cores and fossil pollen and ocean sediments and lake sediments, coral, stalagmites. And there's some historical data that we think of as proxy, maybe ship records, things like that. These are documented. There are specialists in this field in each one. Paleoclimatology is the field where people are studying these climate proxies. So physical, chemical, biological markers that are preserved in the environment. And they're preserved that allow scientists to understand how our climate has varied over thousands to millions of years. Here are even more examples of proxy data. And I'm looking at this website, the National Centers for the Environmental Information, and it has proxy data in it. So if you want to check it out, this is a great website. Hopefully it is still full of information and up and running. Some examples of proxy presentations students have done are plant macrofossils. So these are preserved remains that are large enough to be seen without a microscope. So these are leaves and needles and cones, stems and seeds. And the analysis of plant macrofossils is usually done in conjunction with pollen, which should be an E, and sediment analysis. Plant microfossils are used as climate proxies because plants track climate closely. And many micro and macroscopic plant remains preserved in sediments for thousands to millions of years can be evaluated. You can identify the plants that were present, evaluate how communities have changed, and then scientists infer past temperature, past moisture, seasonality, and disturbance regimes. So we think of proxies that are what things that used to be alive, like plants, as biological sensors. And plant fossils are preserved usually in mud or sediments. Plant microfossils, macrofossils could be pollen, spores from ferns and mosses, um, phytoliths, so that's silica bodies in plant tissues, algal microfossils like dinoflagellates and green algae, and even stomata. And plants make really good climate proxies because they're constrained. They can only live in certain temperature thresholds with certain water availability, and they have growing seasons. We can see CO2 concentrations sometimes in their stomatal traits. So when the climate shifts, plant communities reorganize. Sometimes we can predict how they would reorganize based on what we see plants doing today. And those fossils preserve that organization. And again, they accumulate in lake sediments and peat bogs and floodplains and in marine sediments, even via rivers and wind. And they're dated using radiocarbon, um, tephrochronology, VARVs, it's called stratigraphic correlation. And these create time series of vegetation change. So pollen and spores can tell us about regional changes. We can infer mean annual or seasonal temperatures, precipitation, biome shifts, potentially from a forest to a grassland. And it's best for 100-year to millennial timescales. And those phytoliths that tell us a lot about moisture and land use. So those are the silica bodies formed inside plant cells that are really abundant in grasses. And they're sensitive to how arid it is, the seasonal rainfall, and C3 versus C4 vegetation. And they preserve well where pollen does not. So, like in the tropics and arid regions. Algal microfossils. We use this in hydrology and in temperature proxies. The species assemblages can indicate like the temperature of the lake, the salinity, the nutrient status, and there's certain plants and algal species um, that we realize now tells us and signals maybe a warmer, stable water system versus cooler, more upturning water system. So lots and lots of cool things that we can see using plant micro macro fossils. And in many cases, they can be identified quicker macro fossils than pollen. They tend to be heavy or found close to where they grew, and they can be used in conjunction with other proxies. So I was just talking to you about both plant micro and macro fossils. Pollen, though, is another climate proxy that I mentioned. So you can use fossilized pollen to learn more about our changing climate. So pollen doesn't always make it to the ideal destination, and it is blown in the wind or moved by animals. And pollen is used as a climate proxy because plants are very sensitive to climate, again, and those pollen grains preserve extremely well in sediments. So if we identify and count fossil pollen, scientists can reconstruct past vegetation, and from that, past climate conditions. So pollen is reflecting climate-sensitive vegetation that has grown in the area, and each plant species produces distinctive pollen. And with different plants thriving under different climatic conditions, you can see temperature changes from the pollen. Is it a boreal versus temperate forest? Precipitation are these wetland plants or grassland plants? Seasonality. Has this been a monsoon area in the past, or is it more of a Mediterranean climate with distinct rainy seasons? If scientists see spruce or fir pollen, they might infer cooler conditions. And oak and hickory pollen, they might infer warmer temperate conditions. Grass and sagebrush pollen tells them this was dry or open landscape. So the climate signal can be encoded in the vegetation composition. And pollen, again, preserves well in sediments. Pollen grains, let me make sure I'm recording. Okay. Pollen grains have an outer wall that's highly resistant to decay. So they can accumulate in lake sediments, peat bogs, wetlands, and in marine sediments. So acting as a timeline for us. So deeper layers represent older or longer period of time ago. Researchers can extract sediment cores and identify these pollen grains under microscopes and scanning electron microscopes, and they can calculate relative abundance of pollen types at each depth and create pollen diagrams where these assemblages, again, indicate if a forest expanded or contracted, appearance or disappearance of climate-sensitive species. Pollen is an incredibly important proxy data for us, and it's especially good at long-term climate trends, post-glacial warming and cooling, shifts in moisture regimes, and biome transitions. There are limitations. There's a transport bias. Not all pollen moves the same distance away from its source. So wind and water can move it away, so you have to be mindful of that. Sometimes you can only identify pollen to the genus or the family. Agricultural land use or agriculture and fire can mimic climate signals when you're looking at pollen. So it's best to use it alongside other proxies like charcoal, stable isotopes, macrofossils, lake levels. So while pollen doesn't record climate directly, it records the plant's response to climate. And because vegetation integrates temperature and moisture and seasonality and disturbance over time, pollen is a very robust and widely used climate proxy in Earth system science. And the way it's collected can be from a sediment core, can be in lakes and rivers, and as I mentioned, at deeper depths on our seafloor. Thanks so much for listening.